Good afternoon, everyone. This is my first poster presentation of my research project, and my research topic is the characterization of the mutational signature of acetaldehyde and formaldehyde to investigate its role in carcinogenesis. So, basically, I'm a PhD student, and I'm doing my research under Dr. Keen Chen. So, I'm picking two different aldehydes, acetaldehyde and formaldehyde, and besides its different properties, these are like highly carcinogenic and both are highly reactive compounds. So these chemical actually causes mutagenesis. So mutagenesis is the process of producing mutations in DNA molecules, and single-stranded DNA are more susceptible to damage and mutations. So these mutation actually forms mutational signatures. So single base substitution mutation forms single base substitution signatures, double base substitution mutation forms double base substitution signatures, and so on. And these different types of signatures, they do not have the radiology. So what we do is we actually compare these characterized signatures to the known cancer signatures, and the database of these different types of somatic mutation that actually forms a mutational signature is Cosmic, which is actually the catalog of somatic mutations in cancer. And so this is an example of that mutational signature, signature 4 and signature 5. And in this figure, the pyrimidine of the Watson and Crick base pair is considered, and only 6 different single base substitution, like from C to A, C to G, C to T, T to A, T to C, and T to G is only considered. So the research question that is actually remains for these two different aldehyde are like, what are these its mutational signatures, and what are the signatures related to the cancer. So the research objective is first is to generate yeast mutants in order to investigate its effect in acetaldehyde. The second one is to computationally characterize the mutational signature created by acetaldehyde in yeast genome. And finally, to investigate the function of acetaldehyde and formaldehyde as a carcinogens by deciphering those characterized formaldehyde and acetaldehyde signatures in cancer data. So for the first objective, we would actually generate mutants to find the effect of the acetaldehyde, and for that thing we actually use yeast, budding yeast reporter strain, and the single-stranded specific mutagenesis reporter strain is used in order to generate multiple mutations. And this one was my preliminary result, which tells that the survival percentage has been decreasing once the concentration of acetaldehyde has been increasing, and the mutational frequency has been increased significantly at 75 millimolar, but you could see that has been decreased at 100 millimolar. My second objective is to computationally characterize the mutational signature, which is actually characterized by the acetaldehyde in the yeast genome. So here we could see like different bioinformatics tools and whole genome sequencing has been used in order to generate the variant calling format file, which would actually help in filtration and in the analysis of specific variants which are actually related to acetaldehyde induced mutagenesis. And we use an R package called mutational pattern, and finally non-negative matrix factorization algorithm is used. My third objective is to investigate the function of acetaldehyde and formaldehyde as a carcinogenic. So for this thing, so we would actually calculate the cosine similarity, and finally we actually matches that with the cosine, those cosmic signatures. And the conclusion of my project is to show the mutagenesis of both acetaldehyde and formaldehyde is to characterize the acetaldehyde induced mutational signature in each genome, and it will also show the distribution of each acetaldehyde signature with the number of mutation in each signature. And finally, I will decipher those characterized signatures in cancer data to see if they have contributed in the cancer development.